this is due to primarily is the depolymerization and repolymerization of the actin cytoskeleton. It's also accompanied by changes at the nuclear level that I don't have time to go into, but it can be a very rapid effect, and and although repeated stress in a mouse or a rat or human takes longer to produce its effects, it is something that evolution has used, probably we think, to protect the hippocampus against being blown out when there are low levels of energy availability and the animal is in this low metabolic state. And we think the same thing may be true in the stressed animal, that it's a protective device against permanent irreversible damage. Because when you stop the stress, the process is largely reversible. We're going to talk then about excitatory amino acids, which, along with the inhibitory transmitters of the GABA system, are very important in maintaining the balance between excitation and inhibition in the hippocampus and in many other parts of the brain. The glutamate is a mediator of this stress-induced plasticity, as you'll see in a minute, and it is important for all aspects of normal brain function. So, for example, in the original studies of restraint stress by colleagues in Cleveland, Marty Lowy, Laura Galt, and Keith Yamamoto, during microdialysis, glutamate was elevated during a restraint stress episode, and this did not occur in an adrenal-ectomized animal, indicating that something from the adrenals was involved in triggering this release. We know, in fact, that it is that there is direct action from work of Marianne Joels, among others, direct action of glucocorticoids to stimulate that release of glutamate through a so-called membrane-associated mineralocorticoid receptor. So here's another function of this mineralocorticoid receptor that also is important for nuclear actions of glucocorticoids. So we knew this, but didn't really understand it until Joel's discovery of the direct activation of glutamate. We can block the stress-induced remodeling by blocking NMDA receptors. So we know that NMDA receptors are involved along with glutamate. And now I think is this from Stewart, showing the normal mossy fiber. This is this complex synapse in the stratum lucidum of the CA3 region, which in a water-based trained animal becomes larger, and there are many more active synaptic zones. But after chronic restraint, it becomes much smaller with fewer active synaptic zones, but it can at least partially recover when the stress is over. So we can see these changes morphologically at different levels. Now, many people here have probably heard of epigenetics, and so any of you graduate students are taking an exam, if they ask anything you don't know, just say it's due to epigenetics. So, but it really refers to gene by environment interactions. But originally, when Waddington used the term in a paper in 1942, it was the emergence of characteristics that weren't seen, obviously, from, say, what a fertilized frog egg becomes as opposed to a fertilized human egg, developing and differentiating and beginning to show the characteristics of different cells and different organs and also of different species. But we know that epigenetics now means gene by environment regulation, or some people say above the genome. And in this diagram, you can see that, of course, there's methylation of cytosine residues on DNA, which alters how those clusters of CPG residues, cytosine and guanine residues, respond and regulate gene expression. There's also the histones, which are involved in either packing or unpacking the DNA when they are modified in ways I will tell you about shortly. And one important thing to remember from all of this that I'll say time and time again, even though I use the term reversal after stress, you can't turn back the clock. That it really, the reappearance of characteristics really reflect resilience and redirection and not true reversal of something that's going on. So let's talk about acetylation of, in this case, always lysine residues on certain histones. And one example that this causes unfolding of the DNA so it can be read and transcribed. And an example of this has to do with a metabotropic glutamate receptor, not an ionotropic receptor. This normally inhibits the release of glutamate. It doesn't prevent it, but it keeps the glutamate from overflowing without an actual action potential stimulation. What we find is that this overflow of glutamate is appears to be involved in depressive-like behavior, and I'll show an example of it later, also in degenerative changes in the brain with aging. So Carla Nasca, who's a postdoctoral fellow in her PhD thesis work in Italy, but also with a group in Sweden, showed that a very simple molecule, natural product you could buy in a health food store that's part of our normal physiology, called acetylcarnitine, actually acetylates histones and leads to an upregulation of mGlu receptors, mGlu2 receptors, which are down in animals that show depressive-like behavior, especially in the hippocampus, and we now know especially in the ventral part of the hippocampal formation. And it acetylates histones. The opposite can be obtained by using histone deacetylase inhibitors, which block the removal of acetate. You can have the same rapid antidepressant effects. Now, acetylcarnitine also can acetylate other products in the cell, cytoskeletal elements, and so there may be other ways that it, and also acetylcarnitine activates mitochondrial function. So these are all possible mechanisms by which you have this rapid antidepressant effect in animal models that show a deficiency of acetylcarnitine to begin with. And whether this is true in human subjects, some humans who have depression is something we're looking into right now. Now, the opposite to this is methylation. Methylation generally causes repression. It causes the DNA to become more tightly coiled. It can't be transcribed. Richard Hunter, who's now at UMass Boston, showed this very remarkable phenomenon. After an acute restraint stress, there's a huge increase in immunocytochemical marker for the trimethylated form of histone H3, which is trimethylated on lysine 9. Different lysine than the one that was subject to acetylation in the previous example. And what he found by using antibodies to trap the DNA that was repressed is that among structural genes it also contained what are called retrotransposon-like elements, which are DNA that comes from bacteriophage historically that's become incorporated into the genome. Humans have a lot of it, maybe 40% of our genome, and no more structural genes, than not many more than C. elegans, but it's the retrotransposon elements that used to be called junk DNA that actually probably determine the nature of who we are. Among there are non-coding RNAs that have regulatory function whose levels are repressed by the acute stress. So this is something that acute stress does. Now, does this reduce genomic instability because transposons and retrotransposons, as I'll show you momentarily, jump around? In fact, the work of Barbara McClintock showed for corn maize the fact that this transposition results in different kernel color. We know that immunoglobulins develop their specificity by some form of rearrangement of their DNA sequence to have different amino acid sequences to make antibodies. So what is the function of this? And we don't really know yet. Is it when you move DNA around, does this then, especially with, as Richard Hunter found that with repeated stress and with aging, you lose this repression of, does this mean the genome becomes more unstable or less stable? And does that mean that some functions are lost, perhaps with aging? And maybe this is something that accounts for age-related loss, for example, of some of the plasticity that I've been talking about. We don't know yet. So back to an inverted U-shaped dose response curve, we see acute activation focusing in this case on adrenal steroids and excitatory amino acids. Acute activation enhances synaptic function, improves aspects of memory, especially things we ought to remember to survive a threatening encounter. But more intense acute stress can actually have the opposite effect, inhibit excitability and impair memory, and stress makes you stupid, very intense stress. And then we have the adaptive plasticity, which I just talked about, neurogenesis and dendritic remodeling. We also have the damage potentiation with seizures, stroke, and head trauma, which Robert Sapolsky showed, among other things, that glucocorticoids make this damage worse. And then there's conditional loss of resilience, where in an aging brain or in mental health disorders, there is a change in structure, and then it does not spontaneously bounce back when the stress is over. There's also the case of brain aging, which I'll come to briefly in a little bit. So I've said repeatedly, you can't turn back the clock. Jason Gray in our laboratory, one of the things he's done is to show that if you apply an acute novel stress to naive animals, to animals that have been chronically restrained for 21 days to cause these dendritic changes, or to animals that have been restrained and allowed to recover, quote unquote, for 21 days, that in each case, this novel stressor the animal is seeing for the first time in its life produces patterns of gene expression which are quite different. So this is the forced swim test in the acute animal, and in the recovered animal, and in the animal immediately after stress. There are windows that are open, so the animal is more responsive for reasons we don't understand to acute stress. But the reason the bars are different is that for the most part, the genes that are turned on and off are different. Yet there is a core of genes like CFOS and other genes that are always turned on or off by the acute stressor, although not always in the same direction. You'll also note that chronic stress produces different effects, that the recovered animal has different gene expression patterns, and that giving a bolus of glucocorticoids is not stress, it produces a different set. Even though we know that glucocorticoids are involved in the stress action, they're not the only factor. And of course, I've already told you that excitatory amino acids are important, and I'll tell you shortly about other things that are also important. Now, people who have studied the human hippocampus have shown that there is hippocampal shrinkage in prolonged major depression, in type 2 diabetes, in post-traumatic stress disorder, where this may be both a risk factor for PTSD, but also a consequence of the chronic stress that results from PTSD, and in Cushing's disease, where there are mood alterations and memory impairment, and there the good news is that if you correct the hypercortisolemia surgically, that generally there's at least partial recovery of hippocampal volume, of mood and memory capability within months to a year. But hippocampal shrinkage has also been reported as a result of prolonged chronic stress, chronic jet lag, lack of exercise, and with chronic inflammation. And on the good side, regular exercise and intense learning that shown to increase hippocampal volume as have certain kinds of antidepressant treatment, of which exercise is one of the best for at least mild to moderate depression. And this example from Kurt Erickson's work with Art Kramer and colleagues at the University of Illinois show that in elderly subjects, elderly couch potatoes, getting them to walk an hour a day, 5 out of 7 days a week on a running track at the University of Illinois resulted in 6 months to a year in increased hippocampal volume and improved memory, and in other studies also improved executive function and blood flow in the prefrontal cortex. So regular exercise has extremely important effects. It's probably one of the most important things that physical activity that people can do to keep themselves healthy. Now, talking about the hippocampus, which of course is involved not only in mood and memory, but also helping to shut off the stress response, we move on to think about the amygdala, which of course is involved in anxiety, fear, and aggression, but also helps to turn on the autonomic stress response and the HPA response, and the prefrontal cortex, which is involved in decision-making, working memory, but also all manner of self-regulatory behaviors over mood, impulsiveness, and so forth. But it also helps to shut off the autonomic and HPA stress response. Now, so what may happen in the architecture and circuitry of these other areas along with the hippocampus and other areas of the brain like the nucleus accumbens that other people have studied, and for that matter in other parts of the brain like the cerebellum, which we haven't really studied yet, can alter the way the brain responds behaviorally, but also physiologically, and how it then regulates the allostasis and leads maybe to allostatic load in the rest of the body. So let's talk about what happens. Well, amazingly, the same stress, while it causes dendrites to atrophy, to shrink in the hippocampus, also in the medial prefrontal cortex, and now it turns out in the medial amygdala. At the same time, the same stress is causing the basolateral amygdala and the orbital frontal cortex to have dendritic expansion, which in this case mean the animal is more anxious, more aggressive, and more vigilant, and perhaps less able to control its behavior because of prefrontal cortex. The medial amygdala has an interesting story having to do with social defeat, at least in male rodents. The prefrontal cortex, of course, as I've already told you, is very important for all manner of self-regulation, and the hippocampus for memory. So it's an unbalanced brain. Now, this may be a good thing in a dangerous environment because that vigilance and anxiety and even aggressiveness may be good for adaptation and survival. But as I said, if it does not change, if there's not resilience back to another state when the stress is over, then there's an anxiety disorder, something abnormal that has to be treated. Now, I've told you several times that adrenal steroids do not just work in the nucleus. They do work there through glucocorticoid response elements, mineralocorticoids also through their own response elements, and sometimes you have MR and GR heterodimers that have their own effects. You also have indirect effects through membrane-associated glucocorticoid receptors that operate through other messengers like Krebs in the nucleus to regulate genes. But glucocorticoid receptors also translocate to mitochondria, and they biphasically modulate calcium sequestration. So at low doses, they reduce free radical production and protect cells from damage. At high doses, they do what Robert Sapolsky described, they endanger cells because they then lead to overproduction of free radicals and damage. Glucocorticoids and operating through mineralocorticoid receptors, as I said, also stimulate glutamate release. But at the same time, they could cause the generation of endocannabinoids postsynaptically, which operate presynaptically through these CB1 receptors to inhibit glutamate release, or in some cells to inhibit GABA release. So you can either have blockade of excitation or blockade of inhibition, depending on how cells are differentiated. And this may not be the end of the story, but so we know from nuclear actions, we've seen now that adrenal steroids have many, many other effects. And these are some of the other mediators we know so far that work together in relation to the remodeling. They include brain-derived neurotrophic factor, proteotropic releasing factor, tissue plasminogen activator, which actually probably stimulates the release of CRF, which can cause spine loss from the work of talibarum. Lipocalin-2, a secreted protein that is also involved in neuronal excitability, but also anxiety generation, and the endocannabinoids, which I'll mention again a little bit later. So what about the amygdala? Well, there's not time to go through in detail, but an overactive amygdala is found in anxiety and depressive disorders. The effective treatment for chronic anxiety, as in a mindfulness-based stress reduction, can actually reduce amygdala volume because if it successfully treats the anxiety. An overactive amygdala and an underactive prefrontal cortex input is associated with early cardiovascular disease, work of Peter Generos and colleagues. And a timed glucocorticoid elevation actually reduces PTSD symptoms, which I'll now tell you a little more about. In our collaboration with Sumantra Chatterjee, who is a world's expert on the amygdala, he found that with repeated stress, this is what causes dendrites to elongate in the basolateral amygdala. He found with a single traumatic stressor, putting an animal into a Capricorn bag where it can breathe, it's often done to get a sample of blood from the animal before the animal is put back in the cage. Well, what happens after this single stress exposure is a day later, there is not an increase in anxiety and an elevated plasmase, but 10 days later, anxiety is increased, and 10 days later, there's an increase in spine density on the basolateral amygdala neurons. Now, working with them to try to prevent this PTSD-like effect, delayed effect, we started to inject animals, and a simple saline injection would prevent all of this if it was done just before the animal went into the bag. And then we showed by putting glucocorticoids in the drinking water that that did the same thing. You actually prevented this. A group in Israel, along with a long history of low port being associated epidemiologically with PTSD, supplemental port in people undergoing open heart surgery, reducing PTSD symptoms after surgery. But in this other animal model by Hagit Cohen and Joseph Zohar and colleagues, including Rachel Yehuda, it turns out that if you take people who've had a car accident, get them into the emergency room, and within an hour give them a bolus of glucocorticoid, it actually reduces the later incidence of PTSD. So again, it's another illustration of the good side of glucocorticoid action, which I tried to emphasize. All the things that it does to keep us alive and keep cells adapting and functioning. Not the bad guy that we're often led to believe when we hear stories about cortisol elevation with some bad stress. Glucocorticoid receptors are found, for example, in postsynaptic densities of amygdala neurons, as shown here in collaboration with Luke Johnson and Joe Ledoux's lab. They also, as I said, activate endocannabinoid synthesis, perhaps through that mechanism. And you can see here that their degradation is very important. With an animal that lacks the ability to degrade these endocannabinoids by knocking out fatty acid aminohydrolase, studies with Matt Hill, who's from the University of Calgary, you see that while chronic stress will cause a decrease in open arm activity and elevated plasmase, that the FA knockout shows no effect of chronic stress. There's also no effect of chronic stress on dendritic branching in the basolateral amygdala, whereas there is this increase that I described. FA inhibitors, as opposed to knocking it out, FA inhibitors may be a better alternative to reducing anxiety than simply taking marijuana, because it's a little bit more selective than on the those aspects of endocannabinoid formation that perhaps are more important, because a lot of marijuana will simply suppress the whole system, and things then do not happen that should happen. So the prefrontal cortex is another very interesting area that has a lot of applications. In a study that was done by Connor Liston and B.J. Casey at Cornell, after Connor had shown the dendritic contraction in the medial prefrontal cortex, they trained students who were on a task in an fMRI scanner, assess their perceived stress, Sheldon-Cohen's perceived stress scale, gave them a task of cognitive flexibility that involved the prefrontal cortex, and showed that higher perceived stress was associated with poorer functional connectivity within the circuit that involved the prefrontal cortex. The good news is that these alterations were reversible after these medical students were out of vacation. So the young brain has resilience. What we've gone on to show with John Morrison is that with aging, at least in the rat, this declines, this plasticity, the resilience factor declines. Dendrites still shrink, but they don't bounce back as fast. That circadian disruption, work with Ilya Karatsorios, has shown that circadian disruption causes dendritic shrinkage and impairs cognitive flexibility. I guess I probably left out the example I said I would mention, which is using a drug called riluzole on the aging brain with John Morrison. Riluzole enhances glutamate reuptake by glial cells. We've shown that we can remarkably slow down brain aging. We haven't yet tried to see how it affects stress responsiveness, but it does. This glutamate overflow seems to be very important for brain aging. So I've said in my introductory remarks that we talk a little bit about sex differences, because in the medial prefrontal cortex, work of Rebecca Shansky, John Morrison, in our lab, showed that in males, while stress causes the shrinkage of dendrites that project to other cortical neurons, this doesn't happen in the chronically stressed female brain. But in the female brain, neurons that are projecting to the amygdala, and therefore having the downstream control over amygdala function, actually expand their dendrites under chronic stress, but they only do so when the ovaries are intact or they have supplemental estrogens. And this no change happens in the male projected neurons of the male that project to the amygdala. So with this remarkable finding, which Becca has gone on to show affects learning and extinction behavior in males and females in a different way, mechanistically, even though their behavior ends up being the same, we suspect that there have to be a lot of other cell and sex differences throughout the brain. And to illustrate this paper studied by Uda Havel's group in Germany gave to men and women tests of emotional recognition, emotional perspective taking, affective responsiveness, and the scoring on these tests was the same. Men and women scored the same. But they did functional imaging in the scanner and found that there was quite different patterns of brain activation in men and women, supporting the idea that there are subtly different circuits, neurochemical patterns of activation, with the generalization that men and women do things more or less equally well, but there are differences in the strategy that they and their brains use to solve these problems. And we know from work that we've done on estrogen receptors as well as androgen and progesterone receptors that in the male and female brain, you find these receptors, in this case estrogen receptors, like we do with glucocorticoid receptors, not only in the nucleus of some cells, but associated with postsynaptic densities, with mitochondria, with dendrites, presynaptic terminals, and so on, where they regulate all sorts of things, very much like glucocorticoids that I showed you. In general, every steroid hormone operates through nuclear receptors, but also there are non-nuclear forms of receptors, sometimes the same gene product that's modified and operating in a different part of the cell. So finally, we come then to this question of individual differences and how they come about. Obviously, the genetic constitution is important. Early developmental events are important, and experiences that we have throughout the lifetime amplify these effects. And so we have this phenomenon called biological embedding, and I mentioned earlier the reactive or context-sensitive genes that Tom Boyce and Steve Sumi have shown increase the negative effect of adverse experiences, but also maybe enhance the positive effects of a positive environment. Sometimes they're called orchid alleles versus dandelion alleles. And then we have the epigenetic modifications, which are even more important and significant. And to talk about this a little bit more, showing a slide from Sonia Cavagelli's work with Martha McClintock, that animals that are from a colony that are shown earlier in their life to be more afraid of novelty, as opposed to animals less afraid of novelty. And this we can find in any group of animals that we look at, mice or rats. There are these spontaneous differences in their behavior. Sonia and Martha showed in this study that the neophobic animals died about 200 days sooner, both males and females. So something is happening. We don't know the origin of it, but of course the work that Levine and Denneberg started, that Michael Meany and colleagues have carried to the new level, showing licking and grooming, maternal care is very important as a transgenerational influence, because you can do cross-fostering of pups from a good mother to a not-so-good mother. And in addition, we know now that it's the consistency of maternal care, not just the absolute amount, and the timing in relation to disturbances of the pup that makes a difference. So this may be one of the reasons for these individual differences that we see, but undoubtedly not the only one. Jason Gray again used Tally Barnes' early life nest disruption model, which makes the maternal care much less regular, and showed that in animals with, and this is just in the hippocampus so far, in animals that have had this early life stress, when you give them a novel stressor as an adult, they turn on or turn off far fewer genes than the animals that have not had this early life stress. There is a commonality between them, but it seems that the genome of the early life stress animals is more restricted in their ability to respond to experiences later in life. So we also, I think the audience is familiar with the fact that in human development, poverty and low socioeconomic status is associated with the development of poor language skills and executive function, especially the prefrontal cortex, effects on learning ability, the famous heart and visually meaningful differences studies having to do with vocabulary, work that's been done by Gary Evans, by Edgar Denise, by Greg Miller and Peter Chen, showing greater helplessness and distress and poor self-regulatory behaviors from growing in chaos, elevated levels of obesity and blood pressure, even in children like young as 9 years of age, and increased cardiovascular reactivity, lasting effects of early life adversity on body fat accumulation and systemic inflammation is reflected, among other things, in poor dental health. Adverse childhood experiences, both abuse and neglect, the risky families notion of outright abuse, and exposure to family chaos, leading to increased rates not only of mental health disorders, but cardiovascular disease later in life and obesity. And from Miller and Chen's work showing that adolescents that have had harsh climate in their life and respond to challenges, stressful challenges in adolescence with increasing levels of inflammatory cytokine production as a function of their early experiences of harshness in the family. So we know it has lasting effects, and we know this also in some of our animal models as well. So what does it do to the brain? Well, we know the hippocampus is smaller in poverty, low self-esteem, and it's related to also a risk for PTSD later. We know the amygdala is larger and more active in depression and anxiety disorders, and as Sonia Lupien showed, larger in children living with a depressed mother for 10 years. We know the prefrontal cortex, as I've already said several times, is underdeveloped with chaos and poverty and early life abuse, leading to poorer self-regulation, which then has its own consequences. And then, again, back to the notion of we can't roll back the clock, this nice review by Neil Halfon and colleagues showing the trajectory across the life course, where you have positive influences and negative influences that determine the trajectory, looking at it this way and also this way, major events in life. So we know that things can be done, even though we cannot always prevent early life adversity. There are things that can be done to change that trajectory, and that's, of course, what I think we're all about, what you are all about as well here. So what are some of the things that could be done? Well, regular physical activity is important. I've already mentioned that. I've already mentioned, but not specifically, the mindfulness-based stress reduction studies of Holzer and Lazar showing that by reducing anxiety, which doesn't happen in everybody, you can also apparently decrease amygdala volume. And then the more general notion of social support and integration, as in the experience corps where elderly volunteers become teachers' assistants. There they show, they form social groups, their health declines more slowly because they're more physically active, among other things. And they also show improved executive function, very much like the exercise studies of Kramer. And most importantly is the notion of meaning and purpose in life, which I think we underestimate for its contribution to, especially as people get older, but at all stages of life, do something that has some meaning and purpose, whether it's in your job or in your private life after work. So looking to the future, I think the whole idea is that this idea of limited brain plasticity that can be directed, opening up a window and then pushing it in the right direction is a very important notion that enables us also not only to change behavior, but also to affect systemic physiology. So what I've tried to tell you then is gene expression is continually changing with experience. You can't turn back the clock. We talk about resilience and not reversal. The glucocorticoids and glutamate show produce adaptive plasticity, but also can produce damage in a biphasic fashion. There are many co-mediators and modulators and diverse mechanisms from epigenetic to rapid signaling, changes in the cell surface of the cytoskeleton. Structural plasticity is not damage and does appear reversible, using the quotes, but that changes with age and in mood disorders. That there are sex differences in brain structure that are widespread throughout the nervous system because we know these sex hormone receptors are also widespread throughout male and female brains. That there are these individual differences that reflect some kind of developmental influences, and then early life adversity is very important, especially for self-regulation and later levels of anxiety and being able to cope with the real world. So this is what I've told you about, and I question, as I said, is can we awaken the plasticity of the brain and help it change itself? Thank you very much. There are wonderful people in the lab. I mentioned a few of their names, but I can show you and talk to you about them. They've done all the work. I'm very proud of them.
We have time for questions.
So, really fascinating body of work. Great job, thank you. And so, specifically with reference to the hippocampal data from Richard Hunter, have you guys looked at the H3K9 bimethylation or other variants in any of the brain regions like PFC or amygdala? And do you know if it's different between dorsal hippocampus versus ventral hippocampus?
Good question. We know that the H3K9 change appears to be unique to the hippocampus. We don't know whether it's dorsal or ventral or both, but it does not appear in other parts of the brain. And that's remarkable in itself. It suggests that something is very special going on with the hippocampus. We don't know other conditions. We don't know about methylation of other lysine residues. There's so many things that could be studied, but that still sticks in my mind as being, a remarkable feature. And as I said, does this mean when you lose this capability, does the hippocampus become less stable? Is that the reason we don't see reversal or recovery after stress in aging animal, and so on and so forth? Some people, like Rusty Gage, think that the retrotransposon story, the rearrangement, actually may be important for diversity of memory, especially in young. So both may be true, but to different extents and at different ages. There, and then we'll go up there. So yes.
There's increasing evidence that defects in glutathione transmission are secondary to defects in GABA transmission due to collapse of the chloride reversal potential. Have you looked at the chloride reversal potential in any of your experiments?
No, that would be a very good thing to do because, as you know, there is excitatory GABA early in the development of even some of the dentate gyrus neurons are responding to an excitatory GABA. It's something that's really very important. It's also been shown to be potentially important in seizure-related things. So we have not studied that. It's a good question. Question right there.
Bruce, really nice talk. You showed a nice story showing that glucocorticoids are involved in the prevention of PTSD or prevention of stress in animals, and glucocorticoids are involved in the formation of memory. Is there any evidence that individuals that are resilient to depression or PTSD have maybe upregulated glucocorticoid function, and that they're just not for memory given as well?
Well, there are different interpretations of that glucocorticoid effect. And one of them is that it affects memory. But of course, the holy grail of PTSD-like research is find ways of wiping out those memories. And as you know, there are studies with adrenergic, beta-adrenergic blockers. There are also now studies with MDMA. There are other ways of trying to manipulate those, which may be very effective in the aftermath of an acute traumatic stressor. But when you have somebody like who's been in combat in Afghanistan, who has been there repeatedly, and as a result of these experiences have also developed other experiences, wiping out that original or whatever, there may not be an original traumatic event, there may be many of them. That's another whole ballgame, and it's going to change the brain in different ways. And of course, it's really a very important question is whether we can do anything to redirect that after all these bad experiences. But with the acute, we think, for example, that with the acute example I gave you, that endocannabinoids play some kind of role in the progressive consolidation, if you will, of that. and that interfering with this in some way, enhancing perhaps the endocannabinoids, may be a mechanism for preventing the development of PTSD, say after that traffic accident. Yes?
I was interested, I wasn't aware of the fact that the basolateral amygdala were put out there. Do you know anything of the mechanism that allows them to become more prominent while others are?
The question is, why are the basolateral amygdala neurons expanding while the, say, prefrontal cortical, even medial amygdala neurons are shrinking? Chatterjee has done a lot of work in thinking about this. We don't know what the switch is, but we know that BDNF plays an important role in both. It facilitates the shrinkage. It also mediates the expansion. But so far, the magic switch has not been identified. He has a review in several papers that you can look up. I can give you references to them, that talking about this. And it's really one of the remarkable findings that this plasticity, where excitatory amino acids are also involved in both. But we don't know how they can go in opposite directions. Yes, back there.
Generally, sleep under circadian disruption or characterized as poor sleep, which would be an insomnia type of diagnosis or exposure. Could you comment more about how you see sleep playing a role, especially in the resilience process?
Well, have you heard about the lymphatic system and cleaning out the brain? I mean, I think now there are all sorts of angles on good sleep. And it overlaps somewhat with the circadian disruption issue. The work that Ev Van Cotter has done over a period of time showing increases in inflammatory response, dysregulation, and all the mediators we associate with allostatic load is certainly an important factor. And some of the work I saw today in your posters, the idea that sleep sensitizes the amygdala also to respond to challenges. Certainly that, even living, by the way, in an urban environment is different from living in a rural environment in terms of how the amygdala reacts. And putting that together with Pete Gennaro's work on prefrontal cortical control of amygdala reactivity and antecedents of cardiovascular disease, you can imagine how over time a sort of allostatic load that develops can lead to this in people who have other susceptibilities. So I think that in answer to your question, I think the whole world is opening up as to possibilities, including the mediators both in the brain and systemically. And then, of course, then inducing, if we can, improved sleep patterns, which is really the challenge, is undoubtedly going to be able to improve things, but as one gets older, certainly personal experience, it's not as easy to do the disrupted sleep is a feature which you have to put up there. Yes?
If I could just follow up on that very optimistic answer, and I'm with you there on the possibilities. Switching to the idea that for exercise and sleep, there's a combination of both the willingness and ability to engage in those behaviors that does seem to promote resilience. So in laboratory studies, if you take them away, you can quickly show negative changes. And in intervention, if you impose them in some, compelling way, you see improvements. But if we take those to the real world, what do you think is the barrier to more people engaging in those? Is it that the people who are willing and able are hardier, or there's something different about them versus other people, versus something within the person that could possibly be promoted?
Well, I'm thinking of the studies that were done on obesity prevention and getting people to go out and be physically active, and when they didn't want to, actually having groups, come on, come out and do it, even if you're not feeling well. If you have peer pressure, that can help. And my own experience is I have 2 dogs. and the two dogs have to be walked. And I have a Fitbit, and the dogs walk me, and I got to do it even in the dead of winter. And it's, that's their job, and I've benefited from it, I think. Yes.
Have you considered the role of meditation on people's epigenetics and improving resilience? Because the EEG experiments a lot of the time showed that meditating brains actually had quite active waves, produced active waves. So would you consider that this meditation plays a role in epigenetics and resilience?
Oh, I think there's increasing evidence. There's going to be a volume from the New York Academy of Sciences on a conference that was done last year, actually, at the same time the Pope visited, that was done on mindfulness and meditation. And there is, as many of you already know, some interesting, encouraging work about brain changes that accompany both mindfulness-based stress reduction and also meditation. And again, it's the question of, again, the same as the question of getting people to do it. And not everybody can do the same thing. And I was saying at one of the posters today that my mindfulness is painting, because if you get involved in doing something, a hobby, you have to forget about everything else if you want to be successful. So that's another way of stop ruminating about things you can't control, and in a way probably meditating in a way as well. Way back there.
From my account, about 10 different time scales in terms of college and lab. From epigenetic processes that are taking place over the course of some milliseconds or seconds,
Right.
and life force processes all that are emerging over some decades. And do you see that we can actually put these different time scales together? So, for instance, how long is chronic stress? And is it different in the rat model than it is in the human model?
Well, I mean, I think in the rat model, we know something about immobilization versus restraint. The immobilization takes less time to produce the same effects as the chronic restraint stress. And of course, there's chronic unpredictable stress that produces a different set of results. In the human condition, I think it comes down, for me, to again, it's the chronicity and the sense of control. And the degree to which one has, support systems and other means of behaviorally dealing with it, material support as well as social support systems. And then, of course, the history that's built on, which is why I like the Neil Healthline review so much in showing these long-term trajectories and then calling attention to the fact, which is really where the epigenetics comes in, that the brain is changeable, that those social scientists who say biology is destiny and can't do anything about it are simply wrong. You can, but we still have to discover what you can do, because if you don't want to wipe out all manner of, what our visual system has taught us to do. Although I don't know if you know the example of amblystoma, that if you take animals that have had a monocular deprivation and you expose them as young adults to binocular stimulation, that you can reverse the lazy eye. And the first way that was done is by giving them Prozac. But then Prozac wasn't the only way. You could do every other day caloric restriction. And that wasn't the only way, because you could put glucocorticoids in the drinking water every other day and get that effect. So here's an example where something that was thought to be immutable can actually be reversed. Now, jumping forward, I named Takao Hench at Harvard, in an experiment, I'm not sure how ever got passed a human subjects review committee, took people who did not have perfect pitch and gave them valproate, which among other things is a histone deacetylase inhibitor, and they were able to drive them towards a more sense of perfect pitch, which then suggests that HDAC inhibitors, that wasn't the only effect of valproate, but might actually be a way, like acetylcarnitine, of actually activating processes, opening a window, and then you've got to push it in the right direction with something. Now, if you don't want to completely wipe out the ability to perceive visual stimuli any more than you want to do a lot of things, motor learning and so on. So how much can we loosen up the brain without destroying our capacity to function? And that's the other question about plasticity, reinduction.
Okay, well, it's been wonderful. Thank you for a really informative and expansive talk.